Now we have seen or we have talked about the mode of secretion of the hormones. We will move to talk about the classification of the hormones. Now in this chapter we are not going to talk about all the hormones produced in the body, but we will mainly focus about the hormones involved in reproductive activity. And those hormones are involved in the reproductive activity can be subdivided into three groups according to their nature. So we will have uh, lipid hormones, we'll have hormones of a protein nature, so the proteins, and we will have the monoamines, so the hormones which are monoamines. The lipid subgroup have the steroids, which is a family of hormones, and the eicosanoids. The protein subgroup we have hormones which are the gonadotropic glycoproteins, somatomammotrophic peptides, the cytokines, and the small peptides. And finally, we have the monoamines. We will see and we will talk about each subgroup separately and in details. Now, in the lipid subgroup of the hormones, when we say lipids, we mean here we are talking about lipid-based reproductive hormones, what we call lipophilic hormones. So we will be talking about hormones having a lipophilic nature. So they are hormones that are which like lipids. Lipophilic means they like lipids. They can bind to the lipids. They are lipid-based, and this means that since they are lipophilic, they will be hydrophobic. So they cannot bind to water. They cannot. They are not soluble in water. Um, However, most of the aqueous fluids in the body, most of the fluids in the body are of aqueous nature. So which means that those hormones which are lipophilic, once they are produced by a cell and they need to be transported through the blood, they will not be able to circulate blood because of their lipophilic nature. That's why those lipophilic hormones normally, once they are produced, be able to circulate in the blood, they should be carried on transporters that we call binding proteins. So they should be carried by proteins called binding proteins, which will bind to those hormones and transfer them from their site of production to their target cells. So always those lipophilic hormones need to be whenever they need to be transported in the body fluids, they should be bound to binding proteins which are transported or carriers that carry them to their target sites. Because in general, the proteins and the monoamines, the hormones which are of proteinaceous nature, are monoamines, um, are more hydrophilic than the lipids. And in general, the proteins are more hydrophilic than the lipids. That's why they can bind to the lipids. We have binding proteins which will bind the lipophilic hormones and will transport them in the blood circulation. Now we have two classes of lipid-based reproductive hormones. We have the steroids and we have the eicosanoids. The first subtype of lipid-based reproductive hormones are the steroids. In the steroids, we have four main classes. We have four main classes of steroids, which are the progestagens, the androgens, the estrogens, and the corticosteroids. Now, all of those classes, they are called steroids. All of those hormones are called steroids because they share a common function, a common chemical function, this one, which is the sterol function. So all of those hormones belonging to the steroids family are they belong to the same family. All of those hormones belong to the same family because they share this sterol function in their chemical structure. So that's why all of them, they are considered as steroids because they have this sterol function in their structure. So we have four main families of steroids, or four main classes, which are the progestogens, the estrogens, and the corticosteroids. Now, three of those classes, which are the progestogens, androgens, and estrogens, are considered as sex steroids, steroids having reproductive function in the reproduction. However, the corticosteroids, they are steroids, but they don't have a role in the reproduction. That's why we will not talk about them in this chapter, or in general, we will not talk about them in the reproduction course. So those corticosteroids are produced by the adrenal glands, which are small glands above the kidney, on the kidneys, on the top of the kidneys. Um, like the cortisol, for example, the cortisol is a corticosteroid. They have a function in the immune system, regulate inflammatory response, and also in the metabolism, the homeostasis, and other functions in the body. But they don't have a role in the reproductive system, and that's why we will not them in this chapter. So the steroids are a large group of molecules. They are all derived from common sterol precursor, the cholesterol. Now we are going to see that all of those, all the members of the steroid family, they have a common precursor. They are synthesized from the cholesterol. And that's why they have the sterol function. The cholesterol is synthesized in many tissues of the body and is an important structural component of the cell membrane. As you know, we have the cholesterol is an important component of the cell membranes and is synthesized in many tissues of the body. Now, as we have said, the steroids are synthesized from cholesterol. So this cholesterol can be either de novo synthesized by the body, which means that the body can synthesize this cholesterol from the acetate. So the body can synthesize this cholesterol starting from the acetate molecule. So from the acetate, the body will synthesize the cholesterol, and then this cholesterol can be transformed or converted into pregnenolone. Either the cholesterol can have a source, a nutritive source, so it can come from the nutrition, and the body will take it following from our diet, and then it will be used for the synthesis of the steroids. So the cholesterol used for the synthesis of steroid can be either novo synthesized from the acetate, or it can be taken from the or nutrition from the diet, and then used for the synthesis of the steroids. Now, the first step in the synthesis of the steroid is the conversion of the cholesterol into pregnenolone. This is a first and a common step for the synthesis of all of the classes of the steroids. So the pregnenolone, the synthesis of the pregnenolone, is a common step for the synthesis and the production of all of the four families of steroids. This step of where we have the conversion of the cholesterol and pregnenolone takes place in the inner mitochondrial membrane. So it takes place in mitochondria, in the inner mitochondrial membrane. So we have enzymes that will transform this cholesterol into pregnenolone. Then this pregnenolone will be converted into other steroids, give the other steroids, which are the progestogens, the androgens, and the estrogens, and also the corticosteroids. So the pregnenolone is a common step for the synthesis of all of the other steroids, the sex steroids, and the corticosteroids. And this step will occur in the smooth endoplasmic reticulum. So here, the pregnenolone, which was synthesized in the mitochondria, will be transferred to the ER, the endoplasmic reticulum, where it will be converted into the other into the different families of steroids. Now here we can see that we have this cholesterol that is converted into pregnenolone, and the pregnenolone will be converted into progesterone. Now, before I explain this figure, let's just say what are the progestogens. Progestogens is a family of steroids which have a major hormone, which is the progesterone. So the progesterone is the major and the most important progestogen. So when we talk about progestogens, we will mainly be talking about progesterone. Now, what are the androgens? The androgens are known as the male hormones, and they are the hormones responsible for having a very important role in the male reproduction and the development of the male reproductive organs. That's why they are called male hormones, but they are also present in the females, but at much lower concentrations. And we have four androgens. There is the DHEA, which is the dihydroepiandrosterone. The DHEA, which is the dihydroepiandrosterone. We have also androstenedione, testosterone, and the DHT, which is the dihydrotestosterone. So we have four types of androgen: the DHEA, the androstenedione, the testosterone, and the DHT. The estrogens are known as the female hormones because they are responsible for the female reproductive functions and the female reproductive development. They are also present in the males, but at very low concentrations, and they are known as the female hormones. So when we talk about estrogens, we have two types of estrogens, the estron and the estradiol. So those two hormones, they belong to the estrogen family, and which is known as the female hormone. And the corticosteroids, here we have the aldosterone and cortisol. They are corticosteroids. Now, all of those hormones, each family of those hormones, why they constitute, why we separated them in such families, why those four hormones, they are androgens, and those two are estrogens, and this hormone is a progestogen. In fact, each family of those steroids share common features. So the androgens, for example, they have common structural features and which will give them common functional properties. And the estrogens, they have common structural features also between them, which make that they are a family. For example, the androgens have 19 carbons, estrogens, they have 18 carbons, the progestogens, they have 21 carbon. So they share each family, the members or the elements of each family, they share common structural features, which makes them a family. And since they share common structural features, this will give them common functional properties. The androgens, for example, are responsible of the development and maintenance of the male characteristics and fertility, as we are going to see this in the coming chapters. Estrogens are responsible for the development and maintenance of the female characteristics and fertility, and the progestogens are responsible for the preparation of pregnancy and its maintenance, and they are also they have a role in the regulation of the menstrual cycle. So each family has a different role. They share common structural features, and they have common functional properties. Now let's go back here. The cholesterol can be either taken from the diet or synthesized de novo from the acetate, and we will have the cholesterol, which is the common precursor of all of those steroids. And this cholesterol will give the pregnenolone. The pregnenolone also is a common step, is a common precursor of all of those steroids. The pregnenolone is converted in ER to progesterone, and then the progesterone can be converted either with aldosterone or cortisol, so to give corticosteroids. Also, the progesterone can be converted with the androstenedione, which is an androgen. So the progesterone can give either corticosteroids or can be converted to give the androstenedione, which is an androgen. Now, once this androstenedione is formed, the androstenedione can be converted into testosterone, then the testosterone into DHT. So it will give the other androgens. Also, the androstenedione and the testosterone can be converted to give estrogens. So we can see here that the estrogens, the estrogen and estradiol, they have they are converted from androgens. So the androgens are converted to give estrogens. So always when we have estrogen and estrogen synthesis, we should have first the synthesis of those androgens, and then they will be converted into estrogens. So the androstenedione will be converted into estrone, and the testosterone will be converted into estradiol, and here we have an enzyme which is in aromatase that is responsible of this conversion. Aromatase enzyme will convert androstenedione and testosterone into estrogen. So here we can see that all the synthesis of those hormones, the aldosterone, the cortisol, the androstenedione, and the other androgens as the testosterone, the DHT, and the estrogens have or had the progesterone as a precursor. So we had the progesterone here as a precursor for their synthesis. But the progesterone is not only precursor. We can have another pathway for the synthesis of androgens and those estrogens. They can be synthesized from DHEA, which is synthesized from pregnenolone. So here they can be synthesized from we will have the cholesterol which will synthesize which will give the pregnenolone, and then the pregnenolone will be converted into DHEA, which is the dehydroepiandrosterone. So this is an androgen synthesized directly from the pregnenolone without passing by the step of progesterone. So in this pathway here we don't have synthesis of progesterone. And then the DHEA will be converted into the other androgens, into the androstenedione, and then the androstenedione will be converted into testosterone, and the testosterone can be converted into DHT, and the androstenedione will be converted into estrone, and the testosterone into estradiol. So here we can see that all of those steroids, they have a common precursor, which is the pregnenolone, and the steroids with the pregnenolone, they have cholesterol as a common precursor. And then they can be synthesized either by passing by the progesterone, by the step of the progesterone, um, either or the androgens can be synthesized directly from the DHEA without passing from by the step of progesterone synthesis. Now, not in all of the cells we will have the same rate of conversion, which means, for example, the females, most of the androstenedione testosterone form will be converted into estrogen and estradiol. However, in the males, we'll have a little conversion, but not all. So the biggest part of the androstenedione and testosterone produced will remain as such, will not be converted into estrogens, but testosterone can be converted into DHT, but they will not necessarily be converted into estrogens. So this figure is just representative to understand which type of conversion or which hormone is converted into which hormone, or the chemical reaction, what substrate give which product. But it doesn't mean that we should have all those reactions accomplished always. Depends on the type of the cells and on the site of production. Here we can see that in the mitochondria we have the cholesterol that will be transformed into pregnenolone, and then this pregnenolone will be converted into DHEA, which is an androgen, and then this DHEA will pass to the endoplasmic reticulum to give the androstenedione to be converted into androstenedione, and then the androstenedione will give the testosterone, and testosterone will be converted to DHT by the 5-alpha reductase by this enzyme. Now the testosterone and androstenedione can be converted into estrogens. They will give estrogens in the ER. This conversion happens through an enzyme which is called the aromatase, and happens in the endoplasmic reticulum. We can see also that the we have also the pregnenolone, which instead of giving the DHEA can also move and have some pregnenolone which can be in the cytoplasm. They will move to the endoplasmic reticulum where they can be converted into progesterone, and the progesterone can be then converted in the ER to give the androstenedione, and then the androstenedione will give the testosterone, and etc. The testosterone will be converted into DHT, and we will have the estrogen synthesis from androstenedione and testosterone in the ER. So here we can see the different molecules. We can see that the androgens, they share common structural features. We can see that the progesterone, they have a small difference between them and the structure of the androgens. And then we can see also that here the estrogens have a small difference, the structure between them and the androgens. So we have different chemical structures and different functions for each one of those families. So they have different chemical structures, small differences, not big difference, small differences in their chemical structures, which give them different functions. But we can see that all of them, they have this sterol function. So That's why they are all steroid, because they share this sterol function. You can see that they share the same function, all of them. Now, as we have said earlier, we have two classes of lipid-based reproductive hormones, the steroids and the eicosanoids. Now we have talked about the steroids, we will move to talk about eicosanoids. So the eicosanoids are lipid-based reproductive hormones, but they are non-steroidal molecules, which means that they are not steroids. So why they are not steroids? Because they don't have the sterol chemical function in their structure. That's why they are not classified as sterol. And since they don't share the same function, the same chemical structure as the sterol, so they have different functions also. We have two types of eicosanoids. We have the leukotrienes and the prostaglandins. Now the leukotrienes, they have role in inflammation, and they don't have roles in the reproductive process. That's why we will not talk about them in this chapter or in the coming chapters. However, the second type of eicosanoids are the prostaglandins, and they have an important role in the reproductive process. In fact, those prostaglandins, those hormones, they are ubiquitously synthesized, which means that they are synthesized in most tissues, in most of the body tissues, including the ovary, the uterine myometrium, the cervix, the placenta, the fetal membranes, but also in other tissues. And they play a role in the contraction of the uterine myometrium during labor. That's why they help in the childbirth process. Also during the menstruations, the feeling of pain felt during the menstruations is due to the production of those prostaglandins by the uterus, which induce the contraction of the uterine muscles, and that's why there is this feeling of pain. So the prostaglandins, they have a role in reproductive process, not the leukotrienes. Now, the eicosanoids are lipophilic molecules. As we have said in general, that lipophilic hormones, uh, uh, because of their lipophilic nature, they can cross the plasma membrane without the need of a transporter. However, because the plasma membrane is also lipophilic, so they can cross through it. However, the eicosanoids, they need a transporter to pass through the plasma membrane. Now, the steroids can pass through the plasma membrane directly, and later inside of the cytoplasm, they have receptors. However, the eicosanoids, they need a transporter to transport them from the outside of the cell to the inside, unlike the other lipophilic hormones and unlike steroids. The eicosanoids are also rapidly metabolized by the body to inactive products, so that's why they have short half-lives. They don't stay for long periods of time in the body. They are synthesized in seconds or half uh, few minutes or seconds following their synthesis. They will be metabolized into inactive products. That's why they have short half-lives, and they are described to act locally. So they are local hormone. They act in a paracrine way. So they act on adjacent cells, um, or they are described to have short passages in the blood. So either they act locally on paracrine, in a paracrine way of action on adjacent cell. They don't, they are not transported in the blood to distant organs or to distant cells. However, sometimes they can pass in the blood circulation, but they have short passages in the bloodstream. They don't migrate to distant cells. Okay, so they are local hormone. They act locally in a paracrine way, or they can pass in the bloodstream, but they have short passages. They are not transported to distant cells, which is different from the steroids um, and other endocrine hormones. So they are mainly paracrine, they are not endocrine. If they are transported, they have short passages in the bloodstream. Now, we have seen that the steroids are synthesized from cholesterol. The eicosanoids are synthesized from arachidonic acid and not from cholesterol. So, precursor of the eicosanoids is the arachidonic acid, and the arachidonic acid itself is synthesized from membrane phospholipids. So we have the membrane phospholipids, which can give the arachidonic acid, and then this arachidonic acid will be at the origin of will be converted into leukotrienes or prostaglandins. Now that we have seen the lipid-based or the lipophilic hormones, we will move to talk about the proteins hormones. The protein hormones are generally produced as a form of pre-pro-hormone. Those pre-pro hormones will be cleaved in the ER, or more specifically in the rough endoplasmic reticulum, to give pro-hormone, and then the pro-hormone will be cleaved to give a functional hormone, and this last step of maturation will be done in the Golgi apparatus. And then this hormone will be secreted out of the cell. So let's see here. Here we have a cell. We have the DNA that is transcribed into a messenger RNA. The DNA of the hormone, let's say, will be transcribed into a messenger RNA, and then this messenger RNA will be in the cytoplasm, and it will be translated at the level of ribosomes adjacent to the rough endoplasmic reticulum. Here those dots are the ribosomes, and this is the rough endoplasmic reticulum. So here at the level of the ribosomes, we will have the translation taking place. So we will have the production of the pre-pro hormone. So those protein hormones will be synthesized as pre-pro hormones. Once they are synthesized, once they are translated, inside of the rough endoplasmic reticulum, this pre-pro hormone will be cleaved to give prohormone. So in the rough endoplasmic reticulum, we will have the synthesis of the pre-pro hormone, and then this pre-pro hormone will be cleaved to give prohormone. Then the prohormone will move to reach the Golgi apparatus, and in the Golgi apparatus, it will be the prohormone will be cleaved with a functional hormone, active hormone, and this active hormone will be packaged in the Golgi apparatus inside of a vesicle, and this vesicle will be released in the cytoplasm. So we will have vesicles containing or enclosing the active or the functional form of the hormones. And then we will have secretory vesicles containing those hormones, and then those secretory vesicles will reach the plasma membrane, will fuse with it, and will secrete by exocytosis their content. They will secrete the hormones present inside of them. Now we have four subgroups of protein hormones, which are the gonadotropic glycoproteins, the somatomammotrophic polypeptides, cytokines, and the small peptides. We will start by talking about the gonadotropic glycoproteins. So you can see that Those protein hormones are called glycoproteins. What does glyco means? Glycoproteins. When we say glyco, this refers to sugar moiety that is attached to proteins. So the glycoproteins are proteins having sugar group fragment attached to them, and thus we call them glycoproteins. And they are called, this class of protein hormones is called gonadotropic glycoproteins. Gonadotropic, gonado means those glycoproteins have an action or stimulates the gonads. So since they have an action on the gonads, we call them gonadotropic glycoproteins. Those gonadotropic glycoproteins are also called gonadotropins or gonadotrophins. And as we said, they are called so because they stimulate the gonads. Those protein hormones, those gonadotropic glycoproteins, are globular proteins. So they are proteins having a globular shape, and they are composed of two glycosylated polypeptides. They are composed of two glycosylated chains, which means two polypeptides, uh, two chains of polypeptides having sugar moiety attached to them. We call them two glycosylated polypeptides. And those two glycosylated polypeptides composing the proteins are chains, alpha and beta chains. That's why those globular proteins are called dimers. Dimers, which means they are composed of two subunits or of two chains. Okay, they are composed of two chains, the alpha and beta chains. And since those two chains are not the same, are not identical, we call those proteins heterodimers. Hetero, which means different, so they have different chains. If those two chains composing those proteins were the same, we'd call them homodimers. Since they are different, we call them heterodimers. So the gonadotropic glycoproteins are heterodimers. They are proteins composed of two different chains. They are composed of an alpha and beta chains, which are two glycosylated polypeptides, glycosylated subunits called the alpha and beta chains, and they have globular shape. So that's why they are globular proteins. So the gonadotropic glycoproteins, they are globular proteins composed of two glycosylated polypeptides or of two glycosylated chains, which are the alpha and the beta chains. And that's why we consider them as heterodimers. Now, we have three types of gonadotropins. We have the FSH, which is called the follicle stimulating hormone. We have the LH, which is called the luteinizing hormone. And we have the CG, which is the chorionic gonadotrophin. Now, the chorionic gonadotrophin is produced by the placenta. The FSH and the LH are produced by the anterior pituitary gland. As we can see here, this is this is the pituitary gland, which is also called hypophysis. So here we have the hypothalamus, and this is the hypophysis, which is also called the pituitary gland. Pituitary gland, if you can see here, is composed from two parts: an anterior part called the anterior pituitary gland or the adenohypophysis, and a posterior part called the posterior pituitary gland or the neurohypophysis. Now, this anterior pituitary gland is called adeno. Adeno means glandular, is a glandular parts that secrete hormones. This anterior pituitary gland will secrete the FSH and the LH. The posterior pituitary gland is called a neurohypophysis because it has neurons inside of it, and those neurons under a certain stimulus, we'll see this later, and produce hormones, which are neurohormones, as the oxytocin and the vasopressin. So this is the anterior pituitary gland, the anterior part of the hypophysis called the adenohypophysis, and this part produces the FSH and the LH. So the FSH and LH are produced by the anterior pituitary gland, and they have a role in the regulation of the steroid and gamete production in the testes and ovaries. As we are going to see in the coming chapters, they have very important roles in the regulation of the production of steroids and in the gametes production. Now, from structural point of view, those gonadotropic glycoproteins, they have a very similar structure to another hormone, which is the TSH. Now, the TSH is not it's not a gonadotropic glycoprotein. It is the thyroid stimulating hormone, and this TSH is not the thyroid hormone. So here we are not talking about the thyroid hormone, but we are talking about the thyroid stimulating hormone. So those gonadotropins are closely related in their structure to the SH, and since they are protein hormones, and as we have said earlier, they are synthesized as pre-pro-hormones in the ER, the rough endoplasmic reticulum, and then transformed to prohormones. Then the prohormone in the Golgi apparatus is cleaved to give a hormone, an active or a functional hormone, which are then packaged into secretory vesicles, and they are secreted by exocytosis. Once they are secreted, and because they are globular proteins, they are hydrophilic. So those proteins are hydrophilic. That's why once they are secreted, they can pass in the blood circulation, and they can be transported by the plasma, the blood, without the need of binding proteins and without the needs of carrier proteins. They are not like the steroids. As we have seen, the steroids, they are lipophilic. That's why to be transported in the blood, they need to be carried by binding proteins. Here, the gonadotropic glycoproteins, because they are hydrophilic, they don't need to be transported by or carried by binding proteins. They migrate in through the blood to reach their target cells, which are the gonads, which are in the gonads. Those cells will be in the gonads. So their target organs are the gonads. Second subclass of the protein hormones are the somatomammotrophic polypeptides. In fact, as their name indicate, those hormones are polypeptides. They are composed from one polypeptide chain. One chain of polypeptides compose them, not chains like we have seen in the now the second subclass of the protein hormones are the somatomammotrophic polypeptides. In fact, as their name indicates, they are polypeptides. Those hormones are polypeptides composed from one chain of polypeptides. They have only one chain of polypeptide composing them, and not chains, not two polypeptide chains like in the case of the gonadotropins that we have just seen. So the somatomammotrophic polypeptides are composed from one polypeptide chain, and if we can see the name also, we have the name, we are saying mammotrophic. Mammo is related to mammary, so the name is related to the mammary gland. When we say mammotrophic, which means they have an effect on the mammary gland, either in the lactation, as we are going to see, or in the growth of those mammary glands. And somato means that they have an effect on many tissues in the body. So not all of those, some of those somatomammotrophic polypeptides will have an effect on many tissues in the body, like is the case of the growth hormone. So that's why they are called the somatomammotrophic polypeptides. Those hormones, they have an effect on the mammary glands and growth of tissues. Some of them, like the growth hormone, have an effect on the growth of tissues. In this subclass of protein hormones, we have three members. We have the prolactin, the placental lactogen, and the growth hormone, which is also called somatotrophin. So the somatotrophin, or the growth hormone, has a role in puberty, since its levels increase, are observed to increase near the puberty, and also the children who have a deficiency in the growth hormone will have a delayed puberty. Okay, so the growth hormone has a role in the puberty, and also, as the name indicates, they have a role in the growth of the tissues and organs. It stimulates the growth of the bones and the growth of many other tissues, and it is mainly induces, in fact, the synthesis of proteins. So it induces the synthesis of proteins and then the growth of the organs and tissues. This is related to the growth hormone. Now, the prolactin and the placental lactogen have role in lactation. When we say lactation, which means the production of milk by the mammary gland. Whenever we see this term uh, like in lactation, or lac here like in prolactin and lactogen, it refers to lactose, which refers to milk. Okay, because lactose is the sugar present in the milk. So whenever we have this term, this refers to milk and role related to milk and milk production. So the prolactin and the placental lactogen, they have a role in lactation, and also the prolactin has a role in the development of the mammary gland. The placental lactogen might have a role also in the proliferation of the cells in the mammary gland, and also in the metabolism of the pregnant woman. The third subtype of protein hormones are the cytokines. The cytokines are small polypeptides composed from one or two chains. So in general, they are small polypeptides composed from one or two chains. And they act mostly in a paracrine and autocrine way of action, and in less oftenly in an endocrine way. So mostly they act in a paracrine and autocrine way of action. In fact, we have a lot of cytokines that are produced in the body. Many of them are produced by the immune cells. But here, regarding the reproduction, we will mainly talk about the inhibins and activins, which are cytokines, and which are produced in the gonads. We will see that, for example, they can be produced by the Sertoli cells in the male, and by the follicular cells in the females. Now, the last subgroup of the protein hormones are the small peptides. As their name indicates, they are small peptides. Those proteins or those hormones are very small peptides. They are produced as large polypeptides. So when they are produced, they are first in a form of large polypeptide precursors. Then this large polypeptide precursor will be cleaved to give the functional form of the polypeptide, and which will be a small peptide. So they will be cleaved. This large polypeptide precursor will be cleaved to give a small functional peptide. We have three small peptides implicated in the reproductive process, and those three small peptides are the GnRH, the gonadotrophin releasing hormone, the oxytocin, and vasopressin. Now, regarding the gonadotrophin releasing hormone, this hormone is produced by the hypothalamus and then released in the blood circulation, um, released in a small circulation between the hypothalamus and the hypophysis, the anterior pituitary gland, and the small circulation is called the hypothalamo-hypophyseal portal system. So it will be released in the portal system between the hypothalamus and the hypophysis. It will reach the hypophysis, and under the action of the GnRH, the FSH and LH will be produced by the anterior hypophysis or the adenohypophysis. Now, the GnRH, the active form, so the GnRH is a decapeptide. So it is a peptide composed from 10 amino acids, because deca means 10. So it's a peptide composed from 10 amino acids and derived from the cleavage of a precursor called the pre-pro-GnRH. So at the origin, the GnRH was produced as longer polypeptides, as a larger polypeptide having more than 10 amino acid, and this is a pre-pro form. Then this pre-pro GnRH will be cleaved to give the 10 amino acid active peptide. So if you consider this as the structure or the structure of the GnRH, the composition in amino acids, so there will be cleavage of this part and then this part, and the remaining part is this 10 amino acid part. This will be the active GnRH, the functional GnRH. The other two small peptides are the oxytocin and the vasopressin. The vasopressin is also called ADH for antidiuretic hormone. They are nonapeptides, so they are peptides composed from 9 amino acids, and they are produced by the hypothalamus, by the neurons in the hypothalamus, but they are released by the posterior pituitary gland, which is the neurohypophysis, as we have already specified. So the oxytocin and vasopressin are produced by the neurons of the hypothalamus, but they are released in the posterior hypophysis or the posterior pituitary gland, which is called the neurohypophysis. Now, the oxytocin then when they will reach, we said they will, because the hypothalamus have neurons, and those neurons extend their axons to the posterior pituitary gland. We have the axons of those neurons in the hypothalamus extended to the posterior pituitary gland. In this posterior pituitary gland, at the nerve termination, neuron terminations, we will have the release of oxytocin and vasopressin, which will pass in the blood circulation following their release. So the oxytocin released into the blood have an action on the uterus and on the mammary gland. In fact, the oxytocin induces the contraction of the uterus, of the muscles in the uterus, and which will, and that's why the oxytocin have a role in labor during childbirth. So it will help during the labor due to its action on the uterine muscles. And also the oxytocin induce the contraction of the contractile cells in the mammary glands, and when it induces their contraction, this will help in the ejection of the milk from the ducts of the mammary gland. So that's why the oxytocin helps also in the lactation, so in the breastfeeding the milk, by inducing the milk ejection from the breast. So the oxytocin acts on the uterus and on the mammary gland during pregnancy and lactation. And the vasopressin, it's called vasopressin. Vaso means vessels, from vessels, and pressin for pressure. So it induces the contraction of the blood vessels, and it is also called ADH, or anti-diuretic hormone, because it has an action on the kidney tubules, and it induces the water retention in the kidneys, and that's why it is anti-diuretic. So because it induces a water retention. Now here we will just go back a little bit to this figure of the hypothalamus and the hypophysis. We said we have the anterior hypophysis, which is called the adenohypophysis, and the posterior hypophysis called also the neurohypophysis. And here we have the hypothalamus. So the GnRH is synthesized by the hypothalamus and by the neurons in the hypothalamus, and then released at the level of the hypothalamus in the in small blood circulation that connect the hypothalamus to the anterior hypophysis, and this small blood circulation that connect the hypothalamus to the anterior hypophysis is called the hypothalamo-hypophyseal portal system, and that's how the GnRH will be transported to the anterior pituitary gland, where and when it reaches the this anterior pituitary gland, it will induce the secretion of FSH and LH, which have a very important role during the reproduction, as we have specified earlier. And also we have the neurons, some neurons in the hypothalamus that extend their axons to the posterior pituitary gland, to the neurohypophysis, and the oxytocin and vasopressin are synthesized by the neurons of the hypothalamus, but they are released at the level of the neurohypophysis, and by the neural terminations of the neurohypophysis, and then they will pass by the blood circulation here to reach their target organs.